Alright, so first things first, you have two varieties of tomato. You got your Rutgers and Celebrity. Make sure you label which is which with tape, so that way you don't mix it up once you take them out of the tubes, because then you won't have any labels on them. You're going to take your spatula, or your scoop, either or, and you're going to carefully remove the plant from the tube without breaking too many of the roots. You see here I broke the roots, but got a couple of good ones here. Take off the excess soil. If you want, you can use your distilled water to squirt the soil off. So you got some exposed roots here. And you guys probably can't see, but some of the roots have galls on them. They're very small compared to what you saw a few weeks ago. That's because the plants that we had a few weeks ago had already been infected for about a month or 2 months. These have only been here about 3 weeks. So just take off as much dirt as you can and go ahead and take a look at the roots. So I got a lot of galls here. Now, once you take the dirt off, write your observations for that. And now comes the fun part. Cut off some of these roots. Now for this demonstration, I'm only going to be using a couple of roots, but for you guys, try to get as much roots as possible so you can have a higher chance of seeing the nematodes. And we're going to put it into 10% bleach for 10 minutes. If you want, you can mix it up a bit just to get all the dirt off. But you don't have to keep agitating it like last time. Just let it sit for 10 minutes. Of course, we're not going to wait that long for this video. But let's say it's been 10 minutes. Now we take another beaker. Put all the roots that you have in there. Hopefully you guys have a lot more roots than I have here. Put it at the bottom of the tray. And try not to squish the roots too much either, otherwise you'll squish the nematodes. You'll come up here and you will then put some food coloring into the beaker with your clean roots. So the bleach was just to get any of the crud off of the roots and also to coax the nematodes into coming out of the cells. Because right now the nematodes are not on the outside of the roots, they're on the inside. So we want to encourage them to come out with the bleach. Next, we want to be able to differentiate between the roots and the actual nematodes themselves. So what we do is we're going to add food coloring just to cover up the roots. So you really don't need a whole lot of food coloring. About that much is fine. You can see it's like maybe 10 milliliters. And then you're going to take this and microwave it back there for less than a minute, or up to a minute. The minute is just so that the food coloring goes into the roots. Now I'll skip that step for the sake of the video. You'll then take a plate, just a clear empty plate, and you will take out your roots from the food coloring solution. Where did my roots go? There they are. So at this point, hopefully all your roots are stained. Then you're going to take a scalpel that you guys have at your desk. Make sure you open it from the side that says this end open, because that's the side that doesn't have the blade. So when you put these away, after you clean them, make sure you put them away in that orientation, not the other way. You can take any of these scalpels, and doing this underneath a dissecting microscope, you can start to scrape away the galls to see if you find any nematodes under there. And then if you see anything, you just write down your observations. If this doesn't work because some of the plants just didn't get infected very well or we used old eggs, we have some roots up here in front that definitely are infected because they have a lot of galls on them. Any questions on that? Pretty straightforward, just kind of tedious work. But it'll be cool when you see the nematodes themselves. Alright, so here we have our tank, our tray, and the agarose gel that's sitting inside. However, before we start loading the gel, we need to first prepare our DNA. So again, you've got your PCR tubes, and you can't really tell what's what unless you label them properly. You can't tell whether the reaction was successful just by looking at it with the naked eye. So what we're going to do is load the gel with our PCR. So you're going to carefully remove the cap without tearing it off too quickly, otherwise you'll start to spill everything. And you're going to take your 6x loading dye. What's cool about 6x loading dye is that with 6x you can add 1 microliter of this for every 5 microliters of your sample. So if we had in our sample, in each of these reaction tubes, 50 microliters, how much of the loading dye would we put? It's 5 microliters, 1 microliter of dye for every 5 microliters of DNA. What's that? 10, right. So you get your P10, and you can put 10 microliters of the dye. You can see the dye is very thick and viscous, so try to be careful. And Bryce, I'm going to use your negative control here. And you just dump it out. You can also go up and down with the pipette, just to try to move, or just to try to mix. If not, it's okay. Once you add your dye to all of them, if you haven't mixed it up with the pipette, you can just cap them and then flick it so you mix everything up. And you can see it turns a nice purple color. Now it's ready to be loaded once you have the dye inside of it. We're going to be putting 10 microliters of each sample into the gel. So to load the gel, Max, this is the part where you're going to have to get close. Take 10 microliters of your sample. And you have your wells here. They may be kind of hard to see on camera. But you want to support your hands or the pipette either on the bench or against the tank. If you lean on the tank, make sure you don't put too much weight on it, otherwise you'll flip the whole thing over. So I would put my finger close to the tip of the pipette so you're not shaking around too much. Because if you go down too deep into the well, or if you start smacking the well, you'll ruin it. So stabilize it first, and then you gotta kind of guesstimate, not guesstimate, but really look at it closely to see when the tip is inside the well. And once the tip is inside the well, you're gonna slowly release the DNA into the well. And you only go to the first stop. You don't go to the second stop because you'll introduce an air bubble underneath the buffer, which will explode everything. Well, not explode everything, but it'll mess everything up. So that would be how we do one sample. Now sometimes with molecular biology, this loading dye is good for looking at things on the agarose gel, but sometimes we want to further manipulate the DNA. So in those cases, we do not continue to add DNA to the entire sample. So for example, if I still want to test the DNA, but I don't want to completely ruin it by adding all this dye to it, what you can do is you can take some of the sample, and I'm going to take, Bryce, some 10 microliters out of your positive control, and I'm going to put it on this called parafilm, on this glossy side. So it's the side that doesn't have writing. And when you put it on there, you can do this on the table, but I'm just showing you guys so you can see. It kind of retains its bubble shape because the paper is very waxy. So if I put 10 microliters of DNA, how much DNA loading ladder would I need? Excuse me, DNA loading dye. 2, right. Because it's 1 microliter of dye for every 2 microliters of DNA. So again, we take our loading dye with 2 microliters. 2 microliters, you can see, is a lot less. And then you stab the, well, not stab, but put the pipette tip right up to it and go up and down to mix the DNA. Now we can take 10 microliters of this right, so now we pick it all back up. There's 10 microliters. You're going to have a little extra left over. That's fine because you had 12 microliters there. With the 2 extra that we added, this only fits 10. And then again, you can load it into the gel. So that's how you load the samples. And you see I did that kind of fast, so I kind of screwed up, but that's fine. You also need to load your ladder so that we can tell the distance to each one. The size of each DNA fragment. For the DNA ladder, you can load 5 microliters. And it's the exact same process, except it already has a dye in it. So just put 5 microliters of this ladder into each weld, exactly how I showed you here or here. Well, actually, you don't need to use the parafilm, I guess. Are there any questions on that? No? Okay.